Well, good morning to East Coast and West Coast both, fortunately, and good afternoon to our European colleagues. And this is a very important session today, which we're going to do. So this is on behalf of Assembly on RCMB, Respiratory Cell Molecular Biology, presenting Emerging Junior Investigator Researcher Forum Spring Retreat. So this is second of the webinar series. We did it in last year, late last year one, which was senior investigators. We decided to have a junior investigator. So here, among the juniors, we'll have the speakers as well as the chair. So I'm just being a chair of the Irfan Rahman University of Rochester, along with my colleague Beata Kosmider. Beata, would you like to say about you?
Welcome, everybody. Thank you for joining us today. My name is Beata Kosmider. I'm professor at Temple University in Philadelphia.
Mareike, please.
Right. Welcome also from my side. I'm the European representative of the chairs. I'm a professor of translational inflammation research at the University of Marburg, and I'm really excited we have a lot of junior investigators here.
We are the chairs here from the lung biology, lung aging group of the ATS of the RCMB. So we have two speakers, and they'll be given 20 minutes, 20 to 22 minutes. And then we'll have a 10 minutes question and answer. They will be moderated by our junior colleagues. And I'm just going to say there are two speakers here, but then you will hear more about them from our junior colleagues. And there will be chairs and panelists, moderators, panel discussion 30 minutes. So total will be 90 minutes. So the next one, here are the names, which are our junior colleagues. So what I'll do here, I would like to ask to introduce, ask Beata to let them, our junior colleagues, to introduce. Please, Beata, go ahead.
Thank you. So today we have two junior chairs of this webinar. Dr. Maja Funk, it's a junior PI at Helmholtz, Munich in Germany. And the second chair is Dr. Delphine Beaulieu, and she has a postdoctoral associate position at University of Pittsburgh. Welcome.
Would you like to say, Maja, yourself to introduce here, please?
Well, as Beata already mentioned, I'm a junior group leader at the Helmholtz Munich, and my research group is focusing on aging and environmental stress in lung diseases. We focus on COPD, and we use mainly airway organoids. So I'm looking very much forward to the two talks today, which are super relevant for our research. Thank you.
Delphine,
and I'm
please.
happy to serve as a chair.
Delphine.
Hi, everyone. Yeah, thank you for giving me this opportunity to be co-chair today for this session. So I'm Delphine Beaulieu, postdoc associate at the University of Pittsburgh. I'm working in the Melanie Konishov lab right now. And my research are focused on the cellular senescence, especially in COPD or age-related lung diseases, and understand those mechanisms in this context. Glad to be here today. Thank you.
Thank you. Thank you so much. So without which we would not be doing this webinar is our ATS assembly officers and our RCMB chairs and our executive committee members. And we would like to thank them for their generosity and for arranging this, including Thera, who is among us, who is handling this webinar series. The recording will be available and to our participants. So with that, what I'll do, I'll go back to the first slide and let our junior colleagues to take over this complete webinar series, first ever. So please, now, Maja, please go ahead with Jonathan.
Yes, so it's my pleasure to introduce our first speaker, Jonathan Baker. He is a research associate with Mona Bafadel at the King Center for Lung Health and School of Immunology and Microbial Science at the King's College in London. And so Jonathan, throughout his career, his research always, or interest evolved around investigating aberrant signaling and inflammatory mechanisms in COPD. He first, or after his PhD, he joined the labs of Professor Peter Barnes and Dr. Kazuro Hito at the NHLI, where he studied the roles of microRNA in regulating sirtuin 1 and 6, and to investigate how these anti-aging proteins function in COPD patients. And then afterwards, he joined the lab of Professor Luis Donnelly, focusing on IL-36 cytokines in COPD. And now he is working on iron homeostasis, and his talk will be about dysregulated iron homeostasis as a driver of cellular senescence in COPD. And we are very much looking forward to your talk, Jonathan. The stage is yours.
Great, thank you very much. And just share my slides, and thank you very much for the invitation to be here today. Is that clear? Can everyone see the slides? Brilliant, thank you. Yes, thank you very much for the invitation to be here today. As Maja said, I'm a research associate at King's College London as part of the King Centre for Lung Health. And today I'm going to talk about iron and its role that it potentially plays in cellular senescence and the consequences of having elevated senescence in iron in the COPD lung. So many of you have seen this graph before, but it's always how I like to start my talks. But we like to think of COPD as a disease of accelerated aging. On the left axis, on the x-axis, we have lung function. On the y, we have we have age. And what we see is our lung function increases as we age, up to around about the age of 24, 25, in which peak lung function occurs. In normal aging, our lung function will then slowly decline as we age. However, what we see in COPD is this accelerated aging or accelerated decline in our lung function. This is postulated to be caused predominantly by cigarette smoke and driven by ROS. And what we've seen and others have shown in the COPD lung is that we get an accumulation of these so-called senescent cells in the COPD lung. And could this potentially be driving this accelerated aging phenotype that we see in COPD? So what is cellular senescence? I'm sure many of you know this, but just to begin with, what we see is that cellular senescence in its sort of basic form, and it kind of has changed, maybe not the permanent, but more a stable proliferative arrest. So cells go into this stable proliferative arrest, and then this is in response to various stresses. This may be telomere attrition, so this normal healthy aging, or in response to stresses. And what we think in COPD, this is oxidative stress driven by cigarette smoke. It leads to irreversible damage to the lung or a cell and induces a senescent-like phenotype. These senescent cells can be determined by markers such as P16 and P21, these cell cycle checkpoint inhibitors, which stop the cell from proliferating, as well as the senescence-associated beta-galactosidase staining, which we can use. And why these cells are important is they remain metabolically active. So they can release these pro-inflammatory cytokines and inflammatory mediators known as the SASP, or the senescence-associated secretory phenotype. And because they can no longer proliferate, we get this loss of a repair mechanism within the lung, as these cells cannot repair damage. So as we said, we've spent a long time understanding some of the mechanisms in COPD that may drive cellular senescence, with oxidative stress activating the PI3K-mTOR pathway, leading to a downregulation of sirtuin 1 and sirtuin 6. We also see an elevation of DNA damage response in the lung, leading to activation of P53 and an induction of P21, the cell cycle checkpoint inhibitor. As well as this, we see elevation in P16, leading to this permanent cell cycle arrest. These cells are anti-apoptotic, so they don't die and aren't removed. And we see increase in BCL2. And as well as this, we see an increase in NF-kappa B signaling, which potentially drives the SASP. And why we think the SASP is important. So the SASP has been shown to be important in a healthy unchronic state, unlike in disease, as having beneficial effects. But what happens in disease, we think, is this chronic SASP can lead to metastases in cancer. It can drive fibrosis and lead to this low-grade chronic inflammation that we see in many diseases and within normal aging. And why this is important as well is the SASP can potentially lead to paracrine senescence and spreading the senescence around the lung. And if you look at the SASP and what's actually released, so these chemokines and proteases and growth factors, it really mirrors the inflammation or the inflammatory profile we see of the COPD lung. And this is why we sort of see of COPD as this disease of accelerated aging and potentially a disease of senescence. And why is senescence important then? So why should we care about having an increase in the number of these cells in the COPD lung? So as we say, this aberrant SASP, this chronic low-grade inflammation, may lead to dysregulated and unregulated immune cell recruitment like we see in COPD. So it might be leading to recruitment of immune cells that aren't needed or disjointed. The SASP contains proteases, which potentially can drive emphysema. And this lack of repair can lead to a loss of repair against injury and also drive a fibrosis. So my work and really where I'm interested in is in the small airways in the context of COPD. So although debated, it is thought of where the disease initiates. And here, for those who have not seen like a small airway in COPD, in a non-smoker, we have this nice, healthy, open, easy to breathe through airway. Whereas on the right-hand side, we have an example of a COPD airway, which has got high levels of remodeling, has mucus plugging, and you can see this high levels of fibrosis. And if we do immunohistochemistry for one of these senescence markers, so in this case P16, you can see these small airways really light up. And you can see here that the small airway epithelium is really highly positive for P16. So when we look at the small airways in total and have a look across different disease, so non-smokers, smokers, and COPD, and do staining for P16 and P21, we see in COPD that we've got an elevation of both P16 and P21 compared to our non-smokers, and P21 showing compared to our smokers. And if we take lung resection tissue and digest and take out our small air epithelial cells and grow them in vitro, we can maintain these features. So we see in the COPD samples a decrease in SIRT1, an increase in P16 and P21 at both the protein and RNA levels. And again, they show these classic senescence markers. So here we have an increase in DNA damage and SA beta gal. So here you can see the green in the immunofluorescence and the beta gal, this blue staining of these senescent cells. These cells show this typical G1 cell cycle arrest, so they can proliferate once and then tear off compared to the blue non-smokers. You can see the reds go through this one division. On our COPD cells, small A epithelial cells are in G1 cell cycle arrest. And again, yes, they show this classical SASP release, increasing these pro-inflammatory cytokines, as well as more SASP-related factors and proteases. So I think where we're at the moment is we've certainly, many of us in the field have characterized these senescent cells and showed that in COPD there's an elevation in the number of senescent cells within the COPD lung, and we all have our own sort of favorite. But really what I think is interesting to now try and understand is what is inducing this and what is the consequence of having these senescent cells in COPD. And I'm just going to give a little bit of sort of a flavor of some of the work we're looking at and trying to understand. So I've had interested in iron and its role that it potentially plays in cellular senescence for a while. So normal homeostasis, you can see iron plays a role in lots of different functions in the cells, such as DNA synthesis, mitochondrial function, etc. But what's really important is if iron is mishandled through Fenton reaction, it can actually drive oxidative stress, so this process that we know to be elevated in COPD. And if mishandled, this iron can really drive and exacerbate this. And predominantly for this talk, I'm going to talk about transferrin-bound iron uptake. But iron is taken and sequestered by cells by being bound to transferrin. It binds to the transferrin receptor and is endocytosed into the cell, where iron is released into this labile iron pool. It is usually stored within ferritin or used within the mitochondria, but if not handled correctly, can also drive this ROS reactions. And it's exported by ferroportin. There are also other iron transport mechanisms which are also involved through these receptors. And we've known for a long time now, lots of work from Suzanne Clunen's group, but iron and from the past has shown that iron is, the COPD lung is iron loaded. Here is an example of some pearl staining in the middle, and you can see that macrophages are predominantly staining for this pearl staining, with C being an example of a COPD patient. If you look at the levels of iron within the bowel fluid, COPD patients have higher BALF. And if you look at this SIMS mass spectrometry data in which you can spatially resolve iron, you can see there's a high level of staining and an increase in the amount of iron in the COPD lung compared to healthy donors. And what I quite like about this image is you can see there's high levels of staining for iron within the airways. And mouse models have shown that cigarette smoke can lead to an increase in this iron within the lung. So here from Suzanne's group, you can see cigarette smoke leads to an increase in pearl staining and iron within the lung. And what's interesting, if you can chelate the iron using DFP, we can actually, this example showed that you can reduce the amount of cellular infiltrates into the lung and total BALF protein, suggesting that chelating iron reduces this inflammatory response within the lung. So it does show that iron is potentially important in disease. As well as this, recent work has showed that senescent cells are also iron-laden, so they have this mishandled iron and accumulate iron within a senescent cell. And what's interesting is, and it's debatable in the field what the consequence of this, but if you induce senescence by using, in this case, irradiation, you can induce senescence markers, and these cells have more predominant iron levels. And then if you treat cells the other way and treat them with iron, you can induce the senescence-associated beta-gal staining. And what happens is, if you give iron into the lung, you can induce senescence and fibrosis. So it all suggests that altered iron metabolism can drive senescence and drive fibrosis. But does iron play a role in the COPD lung? And senescence play a role in the pathogenesis of COPD? So what I did is first look for these markers in lung homogenate samples. So these are pieces of COPD lung or non-smoker, and then gold stage 1 and gold stage 3 and 4. Lung tissue, here you can see they showed increased P21 and decreased SIR2 and 1. And we also see increased levels of transferrin receptor, so the way in which iron is endocytosed into the lung, into a cell. And also a decrease in ferroportin, so the iron is potentially getting trapped within the COPD lung. So what I did was do some graphophobic atomic mass spectrometry and measured iron in our COPD small air epithelial cells and showed that they had elevated intracellular and heme iron. And what was interesting also, if you fed them iron, so this ferric ammonium citrate, we can treat these COPD and non-smoker cells with. If you look at the COPD cells, you can see they took up more iron compared to the non-smokers, so have a higher capacity to take up iron. And this may be because our COPD small air epithelial cells have elevated expression of the transferrin receptor and ferritin, so a storage protein, and this correlated with senescence markers. What potentially drives this iron uptake is not really unknown, and this is something that we're working on at the moment in the lab, but hydrogen peroxide, so oxidative stress, increases iron uptake into cells. And it also increases the expression of the transferrin receptor, ferritin, the storage protein, and mitoferrin, which takes iron into the actual mitochondria. And what happens if we try and treat cells chronically with iron? So here we can see it sort of reduces our mitochondrial function and drives mitochondrial ROS. So treating ourselves for 7 days with this iron really induces them into a really dampens their mitochondria and makes them more leaky and more reactive, release more reactive oxygen species. And in doing so, does induce a senescent-like phenotype in these cells. So you get an increase in the cell cycle checkpoint inhibitors and inflammatory markers. And what's quite nice is in this model, if you treat if you induce with a senescence-like phenotype with hydrogen peroxide for 72 hours, but chelate iron during this time, you can kind of stop the induction of senescence. So DFO is an iron chelator. And if we put that in during the differentiation process, so when we're treating these cells with the senescence inducer, we can kind of prevent senescence from being induced, suggesting that iron is important in senescence induction. And then vice versa, if we take precision cut lung slices from COPD subjects and treat them with a senolytic, so in this case, navitoclax, here we can reduce our senescence markers in the gene expression, so P16 and P21, and also reduce the transferrin receptor. So hopefully, probably what I've hoped to sort of show you in a real rough way is basically that iron is mishandled in COPD, and senescent cells and maybe potentially small epithelial cells have this elevated iron and senescent features, and that iron may be driving this process. But finally, just for the end, we want to understand the consequence of having these senescent cells. So we've known for a long time that COPD subjects have, when challenged with rhinovirus, end up with a higher viral titer, so they clear the virus much slower, and have increased neutrophilic inflammation, so sputum neutrophils and sputum IL-8. So this is data from Seb Johnson's group where they treated COPDs and controls with rhinovirus. So we've known this for a long time, but not really fully understood why COPD patients have this altered effect with rhinovirus. And what we'd shown in our lab have shown that older individuals, if you challenge them So we take airway epithelial cells and differentiate the air-liquid interface, and then challenge the rhinovirus. Older individuals have a slower viral clearance again, and they show reduced antiviral and viral recognition proteins like MDR5 and have reduced interferon alpha response. We can recapitulate what Seb showed in humans and affect alleles and show they have a higher viral titer at 72 hours. And what we've shown is that these COPD subjects' alleles displayed increased senescent markers, so they showed more P16 expression. So it suggested maybe senescence may be playing a role in our viral response in COPD. So to understand in a clean way what senescence may be playing, we can induce senescence using chemicals. So this is using etoposide, so we can induce a senescent-like phenotype, increasing P16 and P21, and then infecting these cells with rhinovirus. And here you can see that, again, these senescent cells have this slower viral clearance, this in the red, compared to our healthy non-senescent cells. And what they showed is these cells are more inflammatory upon the virus infection, so you see more CXCL8, but less antiviral, so they've got less interferon lambda being reduced and potentially less CXCL5 and CXCL10, so there's an altered response to the virus. And trying to understand what this might be and how and why, we looked at a load of antiviral ISGs and viral response genes. So in the senescent cells, what we saw was actually there was an increase in RIG1, RIG-I, sorry, no real change in MDA5, but actually IRF7 may be downregulated, and this may lead to a downregulation in the interferon alpha that we see. But what's really quite interesting as well, and maybe a mechanism for these secondary, so sorry for why frequent exacerbators may exacerbate further, is if you infect a COPD allele both with HRV and RSV, and here you can see that 10 days post-infection, if we do a Western blot and look for our senescence markers, after they've been infected with a virus, you get an increase in the level of senescence markers. Interestingly, if we look at then the gene expression, so we can hear in this case P16 for RSV, ferroportin was significantly downregulated in the RSV challenged allele cultures. And what's interesting in this that may be blocking iron out being exported. But in HRV-infected alleles, we get an increase again in P16, but the transferrin receptor went up, but no change in the ferroportin. And what happens is if we use a senolytic, so in this case it's dasatinib and quercetin, another form of senolytic, and clear senescent cells prior to a viral infection and then infect them with rhinovirus, we get a lower viral titer, and we don't get this induction in the expression of our senescent markers, or this induction of transferrin receptor or the ferritin. So it's an interesting observation. So as well as undergoing viral infection, what we also see is COPD patients undergo these secondary bacterial infections. So here, work from Visha Vajika's group, what we can see is after a viral infection in humans, what happens is later, after about day 14, we get these secondary bacterial infections. So the exacerbation is primarily caused by a virus, and then we get these secondary bacterial infections. And what's seen is that in frequent exacerbators, bowel levels of iron is increased. And what's interesting is upon early viral infection of allele cultures, iron can be released. And we also know that macrophages are defective in their ability to phagocytose bacteria in COPD from work from Louise Donnelly's group here, showing that COPD macrophages have reduced phagocytosis. So could iron being released from upon viral infection, could iron be released from our airways and affect our macrophage function, reducing this secondary, reduce this ability of our macrophages to phagocytose or actually change their function? So what I did was take some senescent airway epithelial cells and infect them with rhinovirus. And here we can see what happens is you get a release into the media of iron, with copper not being released, just using that as a bit of a control. And if we take on a monocyte-derived macrophage model, so take monocytes from both healthy and COPD individuals, in this case just healthy, and differentiate them in this iron for 12 days in GM-CSF to make a more inflammatory macrophage, and M-CSF to make a more resolving macrophage, and do this in the presence and absence of iron. What we find in these GM-CSF macrophages, and you stimulate them with LPS, if they're differentiated in iron, they become more pro-inflammatory. And if you differentiate them in the iron with the M-CSF as well, they become less they can produce less IL-10, so they're really switching towards this more inflammatory response. And this leads to a reduction in phagocytosis, and therefore these macrophages, if differentiated in iron, cannot phagocytose. And then finally, to make this a little bit more relevant in our directly back into our lung, we can get tissue macrophages from our lung resection tissue. And what we can do is put these down in max column. So simply just using the magnet to try and pull out our iron-laden macrophages. And here, hopefully what you can see is when we put the cells through this max column and see what binds to the magnet. So this is what comes through the flush through. So these are purified tissue macrophages. We then put them through the max column. You can see here, they're quite a nice, clear, less dirty color. Whereas here, if you look, these are the macrophages which bound to the max column when we plunge them through and remove them. And you can see they're fairly full of gunk and fairly horrible. And these macrophages are more inflammatory and sort of mimic what we see with the monocyte-derived macrophage. They release more proteases at baseline and more CXCL8 and IL-10. So trying to put this all together into a sort of a process, a working hypothesis, as it were, what we sort of see is that the COPD airway, small airways, appears to be senescent. And if we infect these, we see a more propensity to this viral infection and a slower viral clearance. They have an altered inflammatory and antiviral response, and iron is released. Now, this iron may affect our ability of our monocytes to differentiate into macrophages or affect our macrophage function, and therefore leading to macrophages which it switches to a more pro-inflammatory phenotype with a reduced phagocytosis. And this may affect our secondary bacterial infections that we see after a viral infection and exacerbation. And with that, I'd just like to thank everyone who's been involved in this work and everyone who has helped me throughout so far. So Mona at King's, Imperial, Peter and Kaz and Louise, as well as Suzanne Cluner at Trinity or Cornell. And thank you very much for listening.
Thank you very much, Jonathan. Really great presentation, super interesting data. So the stage is open for questions. Do we have questions? So you can either raise your hand or put them in the chat. Maybe until the people have formulated their question, I would probably start. So I was wondering that if you use senolytics and then you have reduced, so in case of the infection, if you use senolytics and then you see that the iron markers go down and also reduced viral load. So do you think there is like a mechanism like signaling between the cells, or do you clear just the cells that have the virus in them? So are the senolytic cells those in which the virus is replicating the most, and by clearing them out, you have the reduced viral titer, or is there also a further antiviral mechanism that is kind of induced?
Yeah, so what we're doing with the senolytics, we're clearing these before we're taking our COPD epithelium that we're saying has got higher levels of senescent cell. We're then clearing these and allowing them to clear for 2 days and regrow, and then infecting them post that. So what we think is that we've cleared the senescent cells, which have this aberrant response usually to a virus. And therefore, what we're thinking is that, by clearing these in the first place, then we're making a healthier epithelium, which therefore has a better viral response. And that's kind of what we're seeing with the reduced viral load.
Okay, thank you. Next is Mareike.
Thank you very much, Jonathan. Really interesting data. So what I'm struggling with a little bit, and not only in your research, right? I mean, we have the same problem. It's really hard to understand what's cause and what's consequence, right? And you said multiple times you showed, I mean, if you put iron, then you induce senescence, but if you induce senescence, you also have more iron. So all of this seems to be a vicious circle. So do you think there's any experiment that tells us what's first or what's driving what? I mean, there's this iron deposition in the lung that drives fibrosis. The question is this anything that's relevant for an in vivo, even patient situation, right? Is there any data from, I don't know, anemic people? Does anything at all that could help us address this?
Yeah, it's a really good question, and one I always get asked, and it's really hard to tell, especially with humans. I mean, I think what it is we know that in the concepts of treating people and helping people, we know the iron's there, we know the senescence is there. So if we can target it and you remove those, then that is a good thing. Whether it's a cause or a consequence is very hard to tell, because, as you say, we don't actually know where the iron is coming from in COPD. Like there is no, we don't know, is it a retention problem? So are these cells retaining it rather than actually there being more necessarily iron in the lung? Is it just that the iron is getting stuck in there? So we know that COPD patients are anemic in many cases, so systemically. So is it that the lung thinks it's actually iron starved and therefore it's retaining all the iron and keeping it, and then that's driving pathology and driving senescence, etc.? I don't know. And yeah, it's certainly something to think of how to best model that, because we know we can drive senescence by giving them iron, but we don't know if the senescence was there first as well. So yeah, it's hard to tell, I'm afraid.
Do you see it with aging, with natural aging? Did you show?
So yeah, there are some studies showing that iron levels increase as we age. So it potentially it could be an aging phenotype, exactly. And definitely when you look in the brain and associations with like Alzheimer's and things, there's heavy metal loading and stuff. So it's there. It's just what causal consequence is difficult to really tease out.
Okay, thank you. Next question is from Beata.
That was a great talk. I have a question for you. If you had a chance to look at senescence markers and intracellular iron levels in control smokers.
So yeah, I didn't show everything. They tend to lie in the middle. Whenever we use smokers, they're always neither or. I mean, certainly. So when we look at the immunohistochemistry data, the smokers sit in the middle. They had higher levels of similar levels of P21 to the COPD subjects, but not P16. And then the iron levels are sort of similar, but not as high. They tend to lie in the middle. I mean, a lot of, especially when you look at the small airways, a lot of the smokers will have small airways disease. It's just subclinical, levels of erosion, and therefore their FEV1 doesn't drop to below 70%. But a lot of them do have small airways disease. So yeah, the pathology is kind of there, and so are the markers, but they're just not quite as heavy as the COPD subjects.
Okay, so there's one more question by Duran.
Yeah, thanks for a great talk, Jonathan. Super interesting data. One question is, when you looked at the macrophages, you looked at the MMPs, and obviously you saw a change there. Have you thought about looking at the ECM and whether there's a potential for, well, what changes occur in the ECM, and maybe if you were to place a healthy or less senescent ECM as a way of recovering or protecting from the phenotype, for example, in the response you see to the virus?
Yeah, no, it'd be really interesting to have a look at that. It's not something that I've done yet. So we're trying to set up some co-cultures at the moment. So putting growing the allele cultures as well as the macrophages in the same well, and then sort of all doing media transfers as well to see if the effects and see if we can transfer the iron across. So yeah, it's something to look at eventually, and we've also put in some fibroblasts in there as well, and etc. But it's not something we've looked at quite yet. But yeah, nice for future work to go look at it and see what it does.
Cool, thanks.
Okay, so last chance to ask now, Jonathan, a question. Otherwise, we have also later the panel discussion
Maja,
where he will be.
I have a question, and this may be a point for discussion. So, Jonathan, about several years ago, people have reported the hemosiderin pigment elevated in patients with COPD, and the lungs of smokers. COPD had a lot of pigments, and therefore it's darker in color due to iron overload. And so what was not known that how iron is bound inside the cells, though now the theory is that senescent cells have more iron, and therefore they have more iron viral load. So, but what is missing here, the puzzle, how iron is in those cells, and what forms of iron binding proteins are those cells, and whether hemosiderin plays any role. So maybe this is a very lengthy question, something to discuss during the panel discussion, to think about that. So that's what I just have a feedback.
Yeah, no, good. Thanks, Irfan. That's really interesting. Yeah, and it's interesting to try and find out what form the iron's in, etc.
Okay. So I put the second slide, Maja, and then please go ahead.
Yeah, so thanks a lot, Jonathan, for the presentation. And great discussion. So I'm looking forward to continue later during the panel discussion. And now the next speaker is Natalia Vanegas, who will be introduced by Delphine.
Yeah. Thank you, Jonathan, for this presentation. So we will continue with Natalia Vanegas' presentation. So Natalia, whom I met in the context of the SENET consortium, which focuses on improving the characterization of senescent cells. So Natalia Vanegas is a senior research associate in the pulmonary aging laboratory with Dr. Ana Mora and Dr. Mauricio Rojas in the Division of Pulmonary Critical Care and Sleep Medicine in the Department of Internal Medicine at the Ohio State University. Natalia received her PhD in biomedical sciences and her master's degree in biochemistry at the National University of Columbia and has done her postdoctoral training in pulmonary research at the Ohio State University. She works in the fields of transcriptomic at the single cell and spatial level using platforms such as Chromium X, Visium, and Xenium in situ, and collaborating with a computational scientist with a specific experience for transcriptomic signatures in the lung. Today she will share with us her work on transcriptomic signature for aging and senescence in the lung. Please, Natalia, we are delighted to listen to your presentation. Thank you.
Thanks, Delphine. I would like to thank the organizer and especially Dr. Irfan. It's my pleasure we're here. And I would like to begin by saying during these last 2 years, I have been learning and working about different transcriptomic technologies and platforms and to provide our understanding about transcriptomic signatures in the lung. Today, I'm really excited to share with you our progress in the transcriptomic signatures and in aging and cellular senescence in lung tissue. So we know that the older we get, the more recurrent is the incidence of diseases in men and women, and in particular of lung diseases such as COPD, lung cancer, and IPF. Data from the last and current century show that the incidence in particular of idiopathic pulmonary fibrosis increases with age. In that, the relationship between aging and fibrosis has been extensively studied using animal models, particularly those induced with bleomycin or murine gamma herpes virus, but the response to the bleomycin-induced lung injury differs significantly between young and old mice. To keep in mind, a 3-month young mice is equivalent to approximately to a 14-year-old human. And for example, in young mice, the injury typically results in a robust inflammatory response followed by tissue repair and resolution of fibrosis. However, in all mice, the response at the same bleo dose is often characterized by the exaggerated inflammatory reaction and an persistence of the fibrosis. In validation, our data also showed that the acute murine gamma herpes virus infection in aging mice resulted in severe pneumonitis and fibrosis compared with young animals. Older mice then tend to have increased levels of profibrotic factors like collagen deposition, which contribute to the development of fibrosis. I'm going to move quickly on this slide, as already mentioned and explained very well by Jonathan, but briefly, one of the known hallmarks in aging is cellular senescence. Recall the well-known definition of cellular senescence as a state of permanent growth arrest that occurs when mitotically competent cells encounter stress or damage. Senescent cells are characterized as viable and metabolically active cells that don't die, and these cells present fissure such as cell cycle arrest with high level of P21, P16, and P53 genes, for example, and also present the upregulation of anti-apoptotic pathway, such as BCL2 family. These cells can release pro-inflammatory factors, or well known as the senescent and associated secretory phenotype SAPs, which can negatively impact surrounding tissues. And finally, in our lab, also, our interest is look for and study more features like senescent associated secretory phenotype as indicator increased lysosomal content. We have another feature in senescent cell, but in general, our lab, our focus is on these 4 features. The cellular senescence is a stress response induced by several numerous triggers, inductor, stimuli, among them DNA damage, telomere erosion, epigenetic factors, and mitochondrial dysfunction. At an homeostasis state, like the joint tissue, senescence is induced by DNA damage and development cues. Senescence initiates a tissue remodeling process by recruiting immune cells through SAS. Macrophages and NK cells clear the senescent cells, and progenitor cells repopulate and regenerate the damaged tissue. However, in aged tissue, this sequence of senescence, clearance, and regeneration may be impaired. In these cases, senescent cells are not efficiently cleared, and the tissue is not regenerated and present tissue dysfunction. This resolution of the damage in these cases involve a fibrotic scar with senescent cell accumulation, inflammatory cells, and fibrotic tissue. This phenotype, this tissue dysfunction, is present in IPF, idiopathic pulmonary fibrosis, which is a chronic disease characterized by the formation of the scar tissue due to a normal extracellular matrix. The IPF lungs present areas of fibrosis alternate with areas of normal lung, and also is a progressive disease from lower to upper lobe. The evidence shows an initial epithelial injury that damages the alveoli due to a combination of several factors such as aging, genetic susceptibility, environmental factors, but also to the accumulation of senescent cells. One of the cell types described in the progression of IPF are the fibroblasts, mesenchymal cells. Fibroblasts have been described as cells that remodel extracellular matrix in response to tissue injury and generate fibrotic scars in chronic diseases. Recent studies have identified diverse fibroblast subsets or mesenchymal subtypes in healthy and injured tissues. However, the origin and functional import of injury-induced fibroblast lineage remain unclear. In the Long Eddy lab, we work on identifying and describing the fibroblast subtype or mesenchymal subtypes of the IPF lung by single-cell RNA-seq data. And for this purpose, we have collected and processed more than 20 samples from all donor or our age-matched control on IPF explant. Also, we have integrated the data and tested, validated, and specific markers gene for each fibroblast subtype already described and on our data generated. You can see here the dot plot describing the marker gene by subtype, for example, pericyte and smooth muscle cells with predominant expression for ACTA2, subepithelial or peribronchial fibroblasts, inflammatory fibroblasts, and the more common in healthy alveolar and adventitial, and finally the recently described CTHRC1 fibroblasts, like the cell, cells responsible for collagen production. And this CTRSRC1 is a primary target of TGF-beta signaling and this pathway that promotes fibrosis by enhancing ECM production and fibroblast activation. We show also the UMA visualization based on the gene expression patterns. Here you can see the profile of the cells with different expression profiles and enrichment of the some subtypes in IPF tissue. And the most important signature is the enrichment of CTRSRC1 cluster here in green. An IPF sample is exclusive for this in this tissue, and also subepithelial fibroblasts or peribronchial fibroblasts. Interestingly, the collagen family signature is highly characteristic of CTRSRC1 fibroblasts in IPF tissue compared to the other IPF subtypes and to control tissue. And you can see here in this heat map, in the green, this column in green for CTRSRC1 fibroblast, the specific expression by fibrillar and network information and fibril associated collagen types. And it's very specific for this subtype in comparison, for example, with a smooth muscle cell or ACTA2 positive cells. The expression these cells don't exhibit a distinct collagen family signature related to lung fibrosis. While single-cell RNA-seq provides high-resolution gene expression data at single-cell level, but lacks a spatial formation. To maintain spatial context and increase spatial resolution compared to the other technologies, we have to use a new platform called Senium in situ. This technology allows us to use FFP tissues or formalin paraffin fixation. Formalin fixation, paraffin embedding tissues, and previously checked by H&E staining and RNA quality. And this new transcriptomic platform is based on in situ hybridization of pre-designed props. Currently in our lab, we have our custom panel with 480 props associated to aging and cellular senescence and IPF. And also this technique use the cell segmentation to associate the single transcript or the transcript expression to single cell, and image-based microscopy detect optical barcoding. But what does optical barcoding? The more simple way, this technique used to identify and quantify a specific RNA molecule within tissue sample. This method involves using fluorescent probes that bind to target molecules, creating unique optical signals or barcodes that can be detected and analyzed by Senium analyzer. This technique enables high sensitivity and precision, and also is capable of signature and base localization, for example, for senescent cells, or in our case, for lung cell types, combining a spatial and single cell resolution. In comparison to single cell RNA-seq, this technique also provide the specific downstream analysis to compare the single gene expression. And obviously, the more focus in this technique is spatial transcriptomics. Let's look at some our results to see how this method can be applied on lung tissue. We show the spatial distribution of the main lineage in the lung tissue according to marker genes, the enrichment of some populations such as immune and mesenchymal cell, as well as the reduction of epithelial and endothelial cell. With this technique, we're able to detect a higher number of cells compared to the other techniques, and we can overlap other images such as post-Senium H&E staining or immunofluorescent images. You can see on the bottom H&E images with the pathological annotation according to the pathologist marking 3 different areas. For us, the more important is the fibroblastic foci, which are known as active sites of collagen production and leading agents of fibrogenesis. Our data show enrichment of the several population in IPF lung tissue. You can see here the clustering analysis. It's very similar to single-cell RNA-seq analysis, but here Xenium provide a more robust and more number of cells detected. You can see here the principal or main lung cell types, and also some population enriched like CTRs RC1 and another like 81 cells decrease and together 82 cells in IPF upper lobe and lower lobe. It's very interesting for us to find the specific signature for CTRs RC1 in IPF upper lobe and lower lobe, and match with collagen 1A1, another specific marker for this population. And the special level also we identify areas enriched with expression of different types of collagen that correspond to the fibroblast foci. In red, you can see here these marks in red to identify these areas by the pathologists, and according to the analysis, the fibroblast foci have the specific signature by CTRs RC1. You can see here the fibroblast foci matching with collagen 1A1 and collagen 5A2, but also another collagen, but you can see the another collagen maybe we see a more unspecified in comparison to collagen 1A1 and collagen 5A2. Also, we found how increases the co-expression of CTRs RC1 and collagen 1A1 and collagen 5A2 in fibroblast foci of IPF tissue compared to healthy sample, and as well as the differential expression of ACTA2 and transcript located in the proximity of the blood B cells, but not in fibroblast foci. If you compare the images between collagen and CTRs RC1, the ACTA2 is very specific, surrounding to blood B cells. And the special resolution, we identify 3 areas according to the gene expression pattern and pathological annotations. Here you can see the normal parenchyma with a specific 3 transcripts, collagen 1A1, CTRs RC1, and ACTA2, and we can define the characteristic alveoli in normal tissue and the expression of ACTA2 surrounding blood B cells. But in IPF lower lobe, we can see the enrichment of the specific areas, for example, for fibroblast foci, enriched with collagen 1A1 expression and CTRs RC1 expression, and also in other areas like surrounding fibroblast foci, when you see the expression between collagen 1A1 and CTRs RC1 are reduced. But when you compare the ACTA2 gene expression, this transcript is more relevant or predominant surrounding blood B cells. We define the specific areas and according to pathology and annotation to H&E staining. In the bottom, we show the collagen 1A1 and CTRs RC1 and ACTA2 quantifications. Here is more clear for you, maybe. You can see the predominant expression of the collagen 1A1 and CTRs RC1 in fibroblast foci in comparison to ACTA2, and the high levels of ACTA2 in surrounding blood B cells. After identifying collagen-expressing fibroblasts, CTRSRC1 fibroblasts and fibroblasts foci, we found increased expression of canonical senescent markers. We evaluated canonical markers such as CDK and 1A, CDK2A and GDF15, or PD21 and P16 and GDF15 in CTRSRC1 fibroblasts, and found a significant increase of cells co-expressing these markers. Here, this is this image from Insignion. Here, the H&E staining, we can localize the specific area, fibroblast foci, is morphological characterized for this architecture, the mix and fibroblasts. But if you can see here, the expression is exclusive for CTRSRC1 fibroblasts. And in the second image, I show the cell segmentation method according to several staining, detecting nucleus, interior, and membrane cytoplasmic. The most important result here is the expression of senescent markers. You can see on the right bar and the bottom, the quantification for CTRSRC1 positive cells, and also positive for P21, P16, and GDF15. Also, we have working with a bioinformatician in our lab and another collaboration with CNET labs. And we working on the new tool called DIPSAS to identify senescent cells and senescent genes and different cell types from single cell RNA-seq data, in our case from FFP lung tissue. This tool is possible to identify the senescent cells. Here in the UMAT, you can see the all lung cell type and a specific CTRSRC1 fibroblasts in green. And after applying DIPSAS tool, we identify the senescent CTRSRC1 positive fibroblasts. And also this tool allows us to identify the candidates of senescent genes or new senescent genes. In this case, for fibrotic fibroblasts or CTRSRC1 fibroblasts, we found 3 on specific 2 emergent genes, and we validate this information from this method with according to GC activity score, which show the enrichment or significant insignificant enrichment the senescent fibrotic fibroblasts compared to normal or non-senescent CTRs fibroblasts. We continue with the validation of this data in Insignion, in situ, or in spatial transcriptomics. We can show, we can see the specific co-expression of these transcripts, and we validated 2 canonical senescent markers, P21 and P16. The white arrows indicate this co-expression in some cells and fibroblast foci. You can see the CTRSRC1 enrichment. Finally, to understand the CTRSRC1 fibroblast signature based on gene expression and how they interact with other fibroblast subtypes on IPF tissue, we hypothesize a possible transition into pathogenic fibroblasts under fibrotic conditions that support the notion that CTRSRC1 expressing fibroblast subpopulation are pathologic. You can see here the different fibroblast subpopulations according to gene expression. Here in the first column, the marker genes per each fibroblast subtype. And also we show the alveoli and healthy tissue or normal parenchyma together to IPF lower lobe. This fibroblast subset may be transitional and developed from alveolar fibroblasts according sonaptors, which provide a niche for maintaining different cell types in injured lungs. And these cells undergo sequential stages of activation and differentiation after lung injury and appear to be the main driver of fibrogenesis. You can see the differential expression, for example, in alveolar fibroblasts, here in the alveoli, alveolar. In comparison here, the alveolar fibroblasts surrounding to fibroblasts foci, and also the enrichment by adventitial fibroblasts surrounding to fibroblasts foci, and inflammatory cells surrounding to fibroblasts foci. And finally, you can see here the exclusive expression by CTSRC1 transcript together collagen 5A2 and collagen 1A1. We identify, or we can define the location per each fibroblast subtype surrounding fibroblast foci, and we can continue to evaluate this specific location in fibroblast surrounding fibroblast foci and inside fibroblast foci. And conclusions, we found a presence of senescence CTSRC1, and also the specific fibroblast population in fibroblast foci is CTSRC1 fibroblasts, and also the findings suggest a transition of fibroblasts into a pathogenic state, potentially contributing to disease progression. And finally, it's clear the combination of single cell and spatial resolution allows detailed examination of the senescent cell within the tissue microenvironment and maintain this context and interactions between lung cell types. In our case, our interest is to study the fibroblast subtypes interaction, and also these senescent or no senescent cells in this microenvironment. Finally, thanks to the team in Lung AG with Dr. Mora and Dr. Rojas, and all bioinformatic team, because this is the team of 4 that I have coordinated over the past 2 years. Thank you for our collaboration, different consortium, principal Senate consortium, and also in our human tissue biorepository. Special thanks to the patients and their families. Thanks.
Thank you, Natalia, for this wonderful presentation. If people have questions, don't hesitate. You can ask on the chat or hands up. Maybe I can start with one question. How do you think those findings can influence the therapeutic strategies for IPF or even for COPD, for instance? So you said you you characterize those new genes in nature for specific focus or fibroblast focus. How do you think that can be targeted for new therapeutics?
Yes, our idea in our labs is study the senescent fibroblasts, like the principal or one of the factors induce the Parkinson's senescence and in the senescent microenvironment. Yes, these technologies also it's possible to identify and targeting these cells, and also in our lab we have another projects about the senolytics with the specific targets, and we can evaluate these specific markers after apply or treat with the senolytics. But of course, the idea with this technology is individualize the the study and per sample, and if you can see different tissues and compare the biology and also the mechanisms. For example, in this case, regulate the IPF. In our case, is our experience, but also in any lung disease.
Okay, thank you. Did you try already to target in vitro or in vivo those cell population, the CTHRC1? Or?
Not yet, but one thing in the bench, one challenge is to isolate the specific population. But we work in that part because the idea is to isolate the different fibrillar subtypes and to define the phenotypic and more transcriptomic profile per each fibrillar subtype. Yeah, this is our, we working on that.
Great. Thank you. Maja?
Yeah. Thank you, Natalia. Very nice talk. I was a bit along the same lines as you just mentioned, to isolate those subtypes or the subpopulation. Did you also consider to culture them and then to actually analyze their secretome? So going a bit from transcriptome to the proteome and then to see what they are actually secreting. So how they could drive the pathology of the disease to induce these foci, for example.
Yes, we have evaluated the secretome from fibroblasts from IPF and healthy, and we compare the secretome. We identify the profile is more proliferative profile than inflammatory profile, and specific for IPF fibroblasts. Our next step, this is from IPF fibroblasts, is the mix of all the fibrillar subtypes, but our next steps is isolate a specific subtype and evaluate the secretome. Obviously, it's possible differentiate the secretome. Maybe it's our maybe will be our finding, but I don't know. Yeah.
Okay, thank you. And I would maybe have one. No, Mareike. I will give Mareike the.
Mareike, please.
You can go ahead, Maja. I mean, it's a quick technical question or technological question. So this is, I mean, super powerful, the technology, but it's also super expensive, right? So we are not all going to be able to afford all of this on a lot of samples. So how easy is it to integrate those different data sets, right? There's a couple of data sets now out for IPF. So have you looked at the other data sets and have you tried to integrate information somehow so we can have more replicates, so we can have like more powerful data sets, all of us together, hopefully?
Yes, for Senior in situ is very novel technology, and but with our bioinformatician, it's possible integrate the data and the for example, from Senior per run is possible to run several samples, and after integrate the data to any comparison or and specifically for it's necessary to do the downstream analysis, for example, the differential gene expression, and also, I don't know, pathway enrichment analysis. But the idea is integrate the data and to make the comparison, because when you evaluate a sample per sample, biological responses is more hard. And conclude, obviously, you know, the biological replicates is considered in the analysis, but yeah, it's possible. And also, we can integrate Senior in situ, I mean, special transcriptomics to a single cell RNA-seq data. remember here, this technique is image-based microscopy, and also is hybridization in situ in comparison to single-cell RNA-seq is sequencing-based data. And Maja,
Please,
So,
Maja,
I don't know,
again.
it's clear?
yeah, just because we were now at the technical question part, I was wondering, can you also infer cellular interactions there? Like you could identify, for example, with which cells these CTHRC1 fibroblasts interact, and then what they could drive in the neighboring cells?
Yeah, and sorry, but here in the presentation is for the time was earlier. But yes, I don't know if you see the transcripts here. In the image and software is possible to identify the transcript and generate the images. But the next step is quantify the double positive cells to find, for example, the CTHRC1 positive, but also if these cells express P21 and P16 and another marker. But the idea here on an image is possible to see the transcript just. But the principal challenge with this expression is the boundaries or the associated transcript expression to single cell. Another challenge is to identify the cells, because we have big cells and large cells and fibroblast foci. This green color, for example, is by maintaining alpha smooth muscle actin staining, and we can identify the body of the fibroblast and related or associated the expression. And this part is the bioinformatician are in charge of this part and the cell segmentation methods, but the assay include the cell segmentation kit to define. Define this transcription.
Okay, thank you.
Thank you, Natalia. We can maybe continue to ask questions and discuss with the panel discussion. Oh.
Okay. Start share. Yeah.
Thank you so much.
Thank you so much to the speakers and the chairs. So now we have a junior investigators panelist. Please go ahead.
So I think we can discuss. We have collected a few questions, and I think the first one, drivers of cellular senescence in lung aging disease. I think Jonathan, you have already mentioned kind of a few causes. What would you think is the most, let's say, latest avenue in this direction? Also, maybe from your research.
Yeah, great. I think everything's going to be disease specific, depending on what we're looking at in relation to drivers. And I think that's probably something we kind of need to understand better. I think, as Mareike was sort of alluding to, we don't know whether some of these things are, a cause or consequence. We know we have these markers and we have these senescent cells, and we have, pathways within them which are activated and maybe driven by, like I say, oxidative stress or cigarette smoke or iron, which one it is, we don't fully know. But I think understanding and using, as we've both shown, disease tissue really is important for those kind of to understand and look at these, and then develop models based on those to try and understand and elucidate which may be driving them, and how then we can try and target these.
Can I comment on this?
Of course.
Because I also say this, right? I think it's disease specific, and some part of me believes this. On the other hand, Is it really? Do we have any indication that it is? Can you think of a single inducer that is really disease specific? So iron, I mean, iron is also, right? I mean, putting iron to the lung induces fibrosis, right? So I don't know. Is it a combination of, is it how much of the one inducer and of the other?
I was going to say, I agree with that, but I suppose we can't with, different diseases. I mean, COPD is predominantly caused by cigarette smoking in the Western world, isn't it? Whereas not everyone who develops IPF is caused by cigarette smoke. So we can't say that is the main cause within the lung, can we? Maybe certain, I definitely think different cell types are more specific to different inducers. We definitely see, as your own work said, like oxidative stress, you put on fibroblasts, it does less, and so it's a different kettle of fish depending on what cell type you're looking at. And I think that's what we've got to really understand and try and look at, and how senescence spreads as well. Is it, one cell type becomes senescent and then switches on other cell types? Is it just that we've got the loss of clearance of these senescent cells? Is that what's happening in disease? It's not necessarily we need to worry so much about the inducer. Is it actually the immune system we need to switch back on? So I think there's lots of questions to answer and look at and try and understand, really. But yeah, I think all of these drivers can sort of cause senescence and cause lung aging, as we say, but different diseases can have more predominant inducers, is what I would suggest.
So the same thing, in other words, this is Irfan, Jonathan, with Mareike question on drivers of senescence. So all these drivers, we know that they cause senescence, and we listed them. We have groups of different drivers. But then once the senescence is formed, cells are senesced. Whether they have a similar, regardless of the drivers, they are the same senesced cells in terms of markers, or they have different phenotypes, which comes to the heterogeneity. So tell us something about the phenotypes of senescence when the senescence occurs in the cell.
Yeah, no, I agree with you, Irfan. I think there are definite, phenotypes or flavors of senescent cells, and I think different cell types will act and respond differently to this process. And I think senescence is a very general term, and it's maybe not. It needs to now, as we get better understanding, and the technologies as we're seeing here, are developing, we can go and then really tease out more about them and understand what's different about these senescent cells, and how they may be regulating it. Is it inflammatory senescence? Is it profibrotic senescence? What are they doing, and how is this driving disease, and how should they be targeted differently? Because we know with some of the senolytic work that some senolytics work on some cell types and do not on others. So there's definitely a different mechanism here that's going on, and that really needs to be teased out and understood and looked at. And I think that's where we're getting to as a field, is trying to understand that better.
I agree. I think in vitro, it's really easy to tell them apart, right? Different kinds of senescence. And then it gets really complicated if you want to look at this in vivo. And I'm thinking if that's something where Natalia can also comment on, right? Because there may be spatial heterogeneity, and spatial location will actually help. And what Jonathan also mentioned will also be very nice to look at if you look at senescence spread, right? Secondary paracrine senescence, however you want to call it. Right, Natalia, did you see anything like this in the tissues that you looked at?
Right. On the we working on that, because it's very hard to identify the senescent cells. This is the first point, because according to our work about some tools to identify the senescent cell, the phenotype, the markers. I don't know if you know, we are several gene lists to identify the senescent cells. St. Mayo, for example, Friedman, and another gene markers, but each study is related with a specific model and is more difficult to transfer or to translate in our studies. But for example, according to DeepCells, our tool in the lab, is possible to identify the senescent cells from single cell RNA-seq. The idea now is to translate the tool to a special transcriptomics on a special location. But of course, is the challenge for us, and define these specific markers according several factors, right? Because in IPF, for example, we have aging, but also we have genetic susceptibility and environmental factors. I think, yeah, it's hard, but it's possible to do.
Delphine, I think if you want to follow up on it, because I was actually thinking about one of the other questions just now, but it would go a bit in a different direction now.
Thank you, but yeah, I wanted to ask you, Natalia, if you checked on your tissue the FRINMA, the Saint-Mayo, or the Senset list, or those kind of signature that we know that they are induced. I mean, yeah, even if they are not specific, maybe for IPF, they may help to characterize a little bit senescent cells in the IPF tissue.
Right. Another comment, sorry.
Yeah.
Is for us it's very important, for example, in IPF in particular, is this population CTSRC1 fibrillous, because recently they had described it, but it's interesting to see it's not only one population become to senescent cells, and the interaction between another population in the lung, and specific, for example, our interest in the fibrillous foci, how we can or can we determine or define this population interact to CTSRC1, and how this specific or particular population origin to another fibrillous subtype, for example, the alveolar adventitial. The theory mark this some transition between or transition state between some fibrillous population. Then it's very interesting to see this bar, right? And IPF is our aim.
Yeah. And did you think to use the, I don't know, younger tissue such as, I don't know, lung cancer patients, and do this kind of spatial transcriptomy to maybe have a kind of trajectory of the cell population to determine, yeah, what is the source where are coming from those cell type?
Right. Not the younger. Our the most comparison in our case is to match control, age match control to IPF, but we have some preliminary data in young tissue and old tissue, and also in IPF about the trajectories. We found the principal subtype population of the origin for CTSRC1 are alveolar fibrillous. This looks like these particular fibrillous are the origin for CTSRC1. But yeah, it's preliminary, but the idea now is to translate the study on a special location and to show this result.
Thank you.
Irfan, do you want to switch to the general points, or?
Yeah, sure. So you think it's over, yeah? Okay.
Okay.
So yeah, we have 6 minutes to go, so that's good. Thank you so much for taking care of the discussion. So any more questions or any suggestions or feedback to us, to speakers or to chairs, please do email us. And so what do we like to say in last few minutes? So again, I'd like to give thanks to the speaker, chairs, and the ATS committee and RCMB executive committee and the chair. So we would like to hear something about the future topics, and that will include how we can increase the awareness of aging, because our group is lung aging and the biology of aging, and so lung aging in particular. So we like to see how the diseases are progress based on either senescence or any other parameters of lung aging or hallmarks of aging. And then we like to hear the emerging technologies. We heard from the Ohio State group, Natalia. She presented very well about the xenium technology, and see any other technologies emerging. We like to hear bioinformatics. We like to hear the updated hallmarks of aging and technological advances of therapeutics, omics, epigenetics, repurposing some drugs, senescence, precision medicine based on senolytic, xenomorphic. So yeah, please do reach out to us. These are just the topics which we think will be timely and important. And we will have the much more discussion in the ATS also. We have a critical age, critical care group meeting, and I can send you the information. I think is on the May, let me see my diary here on May 19 on Monday at 11:45 a.m. So we'll have a more discussion on these topics. So what is any other feedback from our colleagues would be welcome to reach out to one of us. So again, I would like to give thanks to the speakers, Jonathan and Natalie, and also our new chairs, Maja and Delphine, and of course, Mareike did an outstanding job, and similarly, Beata unable to stay because of the conflict. And again, this is a group from us, but this is by you to us. So this is a group we are working for you and the ATS committee members. Any members would like to join, please email me and you will be most welcome and reach out to us. So I think with that, we are within the time frame, 12:27 Eastern time. We'd like to conclude this session. Anything you'd like to hear from Mareike? Please, you are there. You'd like to tell us something to conclude this?
I mean, thank you, everyone who stayed. Thank you, everyone who presented and shared. I just want to add one thing. So we've talked a lot about senescence, and we obviously do love senescence, but we also welcome every other aging-related topics, right? So,
yeah, I would like to say thank you because this is the good platform, for example, for us and the trainees to show our work and to express our question, comments, and yeah, thank you so much.
Thank you. That's very nice feedback. Thank you very much for doing this. This was a very nice session, fantastic science, very good discussions. So
good.
And finally, thanks to Tara. She's here. Tara would like to say something to conclude.
Thank you to everyone who came to the webinar, and thank you to all the moderators and the panelists. Y'all did an amazing job, and we appreciate you taking time out of your busy days.
Thank you. With that, we conclude and end the webinar for junior investigators. Thank you so much. Okay, bye-bye.
Thank you. See you soon.
Thank you. Bye-bye.
Thank you.
Have a nice day.
Bye.
Bye.